Oh, be right with you. Hang on one second. I'm just comparing this one last thing here. Okay. Nice of you to join me here. This is my little lab, and just doing a study on mandibles. Anyway, I wanted to do another video that sort of came off of the other video, which was the ecology video, which I want to describe a couple things and explain a couple things as to what I'm doing and why I'm doing it. In that video, I specifically mentioned that we were tracking a new form called Comeditum. Comeditum has worked itself out to be something much better than a species, much better than it could ever be as being a species, because it answered and unlocked a lot of interesting questions that have been asked for a long period of time. The reason I wanted to bring that to mention was because I'm now tracing a new form, and it's sort of difficult because I'm going to, in this little laboratory next year, I'm going to rear several thousand insects, and things that don't fit into an exact pocket, what I do is I give them a temporary name and a potential form, which is potential species, until I can prove what they are otherwise. So right now I'm actually tracing something else, and I want to kind of introduce that. And right now I'm holding this under the name of Eloquentum. I truly believe that what it's going to turn out to be in the end is it will be the darkest version of true Ohioans. I'm seeing a lot of commonality between the two, but there are some differentiation things about it that are distinctly unique that nothing in the genus has. For an example, Eloquentum is showing itself to have 2 trachea in the 7th gill. All other in the genus all have 1 trachea. This also has other mouth parts that are distinct and unique to each other as well. So for now, I'm holding Eloquentum as a potential form. I only have a female that I collected back in July, and I have 3 larvae that I've dissected the mouth parts out of. And so far, with the 3 larvae and the 1 female, they certainly hold up as something distinct. But like I said, I suspect that it'll probably end up being another variation of true Ohioans. And bringing that to light, Comeditum, you can see here I scratched out the word Comeditum because this used to be Comeditum. Comeditum got called Comeditum because all the markings on the body are all comma shaped, and including on the face are all shaped in commas. So I named it Comeditum as a potential species, potential form. What it has actually worked out to show is that this illustration is built on samples that I've collected and Dr. Trevor's 1935 description for Ohioans. If I look at this, they obviously look nothing like each other whatsoever. What happens here is the larvae have adopted a camouflaging pigmentation trait to their maculation to modify themselves to their environment. The adults of Comeditum and Ohioans are almost identical, and so close that I could not possibly hold Comeditum as a potential species. I had to place it into Ohioans. But the larvae were still incredibly distinct. What I ended up finding out is that this sample comes from a dark substrate where the rocks are very black, like you see in that video, because in that video this is what I was chasing, was this form or variation of form, and not this one. This one comes from that dark background. This one comes from 3.9 miles downstream, where the stream becomes meandering and it becomes red clay silt with high chlorophora levels of algae. So anyway, you can see that there is certainly relationship between all 3 of these, but yet all 3 are distinctly different. But yet the adults all have great similarity to each other. While reviewing the others in the genus, because of this camouflaging trait, I decided to start looking more closely at everything else, and it turns out that Conjunctum also has this same camouflaging trait. This variation of Conjunctum comes from a darker substrate This variation comes from the lighter substrate, both the same locations as these. I have also found this now in Magus. So 3 forms are showing that they have different types of camouflaging traits for specific environments. This is very interesting because what we've always thought that, well, one was a hybrid, that this was a hybrid between this and something else. And the hybrid thinking is a great concept, and it still in some places makes sense. But completely, we can see, and I have been able to see from not only reared samples, but reared samples that have been dissected through the exuvia and through matching larvae, and the adults, matching them out to the adult historical profiles, that they are in fact actually the same thing. They are slightly different variations, and it's a genetic profile that these specific insects are carrying. And for here, for an example, is something that I want to touch base with here, because we're going to talk about taxonomic confusion. Taxonomic confusion is a very important part of why Stenacron are so misunderstood. Here's an example. This is the very first time in history that this has ever been seen side by side, and that's what my book is all about, is so that you can compare apples and apples instead of apples and oranges. Everything is on an equal playing field. You will notice that both of these two are extremely similar to each other, but yet you can see distinction within the two. Not only is there a size difference, there is also a maculation variation. And there is, because these are two completely different forms. This is the one and only real true interpunctatum se. This is the real interpunctatum se. This is canadense. This is not interpunctatum se. Part of the reason it's so yellowy orange is because this is actually an illustration on a female, and of course the eggs would be present and they're yellow in color. Both of them have ventral markings that are extremely similar and are very difficult to tell apart. The whole idea of this book is to shed light on this so that people, biologists, and anybody with an interest can quickly separate one from another just by using the illustrations. Another thing that I want to talk about with canadense is canadense has been known as having spiracular spots. What are spiracular spots? If you look at the adult, this is the abdomen of male adults. Both, one is majus and the other is proximum. You will notice that there's these black spots here on the side where the line comes over, on the side, the lateral sides of the abdomen. Those are called spiracular spots. Canadense does not have spiracular spots. The only way it became having spiracular spots is because in 1947, Herman T. Speight synonymized Ohioense with canadense. And he wrote a new description for canadense, including Ohioense. Now, if we look here at this, you can see here's the five here that make what I call the hypothetical complex of frontal, which DNA is actually supporting. So I suspect at some point in time that frontal, being the oldest of all of these, other than canadense is slightly older, that there will be likely be a frontal complex in the future, and that will consist of Ohioense, frontal, proximum, conjunctum, and majus. All of these are the most confused forms in the genus, and part of the reason they're confused is this taxonomic confusion that I'm going to talk to you about here, is if you look very closely at the Ohioense and you look at the canadense, you'll notice that the canadense has also been separated by a dashed line. You will notice that there's no black spots. There is dark black shading on the sides, but there is no specific black spots. Canadense became known for having spiracular spots after Speight synonymized them together and put the two of them together and rewrote the description. So there are a lot of changes going to happen with Stenacron, and part of that is because of this book. But at any rate, there are some things that are going to happen and some changes, and I wanted people to understand what these things are and what it means. Another interesting thing that we're going to talk about for a second is the marks. If you look at this head capsule, this was for Comeditum, but now it's Ohioan's dark form. If you look at this, you'll see all these black spots, like these big black comma marks. That's how it got the name Comeditum, because of those big black comma-shaped marks you see. All of these marks that I'm going to point at, this one, this one, this one, these ones on top, this one here, all transcend into the adult. Believe it or not, you can identify all Stenacron larvae without dissection to what they are going to become as the adult. Stenacron are actually incredibly reliable in maculation, probably well more reliable than Macaffertium, which is very interesting because most people think they're kind of all the same and they're unreliable, and that's not the case. The only thing on Stenacron in maculation that is unreliable is this one little specific entity, which is the median line. Not the submedial lines, which are the surrounding lines. It's the center dark median line. And why it's not necessarily that it's variable, it's because the digestive tract runs right underneath of there. So it is very hard to read that line on Stenacron and read it accurately unless the digestive tract is completely empty. Because Stenacron actually have a diet of mineral content. So they eat a whole bunch of little tiny dirt and rubble and minerals, and it becomes dark in their tract, so it's very hard to read that. But all of these other markings are reliable to form, and form validates species. You can't even have species without form. The other thing is I wanted to make a mention of is that none of this is possible without this. I'm very fortunate that I happen to have one of these. Not very many people do have one of this in particular, of this specific one, but this is my pride and joy, and I could not do this without this book. This is the Biology of a Mayfly, and it is the 1935 edition. And in actuality, interestingly enough, you will notice that the printing is crooked on it. If you notice, it's all crooked set. That's because this is one of the very first 200 issues ever made, and then they've reprinted a first edition. So this is actually a first of a first edition, and it was completely unnamed and unused, and I am the very first owner of this, which I'm grateful for. But like I said, what I'm doing in Stenacron couldn't be done without ownership of this book. So anyway, I hope this gives anybody who's interested in Stenacron a little bit more insight into Stenacron taxonomics and Stenacron confusion. Anyway, welcome to my lab, and thank you for watching this video.